Okay, let's talk about pentose phosphate pathway or PPP. It's a cytosolic pathway that branches off from glucose 6-phosphate. Pentose phosphate pathway is also called hexose monophosphate shunt or HMS. Okay, so it starts from glucose 6-phosphate. Where does this glucose 6-phosphate come from? Glucose 6-phosphate comes from glucose phosphorylation, glucose to glucose 6-phosphate via hexokinase in most tissues or glucokinase in liver or pancreatic beta cells. And another way that glucose 6-phosphate comes from is glycogen breakdown. And another way is gluconeogenesis in the liver. It also produces glucose 6-phosphate. Okay, back to pentose phosphate pathway. The pentose phosphate pathway PPP, its whole purpose is to make two key things: to make NADPH and to make ribose 5-phosphate. To make NADPH and to make ribose 5-phosphate. How is NADPH used? It's super high yield. NADPH is mainly used for building and protection. For protection, it keeps glutathione in the reduced form so cells, especially RBCs, can neutralize oxidative stress. And it is also involved in killing microbes. It powers the respiratory burst in neutrophils or macrophages via NADPH oxidase to make reactive oxygen species. And also it is very important in biosynthesis of fatty acid, cholesterol, and steroids. And also it involves in detoxification or drug metabolism by supporting cytochrome P450 enzymes in the liver. Okay, now we know what does NADPH do. Now let's talk about ribose 5-phosphate. Ribose 5-phosphate is used for building nucleotides for DNA and RNA. Very important in rapidly dividing cells. Okay, back to PPP. PPP, pentose phosphate pathway, has two phases. One, oxidative phase, which is irreversible, and two, non-oxidative phase, which is reversible. Alright, let's talk about the oxidative phase of the PPP. This oxidative phase of pentose phosphate pathway is the irreversible part. It is irreversible and it has one main job, which is to make NADPH. The pathway starts with glucose 6-phosphate, G6P. Where does this G6P come from? It comes from glucose via hexokinase in most tissues or glucokinase in the liver and pancreatic beta cells. Or it can come from glycogen breakdown. So glycogen becomes G1P and then it becomes G6P. Okay, now the oxidative phase begins from glucose 6-phosphate. Step 1. It is the rate limiting step which makes NADPH. The enzyme here is G6PD, glucose 6-phosphate dehydrogenase. The reaction is G6P becomes 6-phosphogluconolactone. What you gain? NADP plus is reduced to NADPH. So this step makes NADPH. Remember, G6PD is the rate limiting enzyme and this is the classic test point. Also remember, if you have more NADP plus, meaning you used up NADPH, the pathway speeds up. If you have more NADPH, the pathway slows down. Okay, more NADP plus, you used up NADPH, the pathway speeds up. If you have more NADPH, the pathway slows down. Step 2. Step 2 is the ring opening step. Here, no NADPH is produced. Enzyme is 6-phosphogluconolactone or lactonase. Enzyme is 6-phosphogluconolactonase. Reaction is 6-phosphogluconolactone becomes 6-phosphogluconate. This step just opens the lactone ring using water. No NADPH is produced here, but the intermediate names are not that important. Step 3. Step 3 is oxidative decarboxylation step, which makes NADPH and CO2. The enzyme here is 6-phosphogluconate dehydrogenase. Reaction is 6-phosphogluconate becomes ribulose 5-phosphate plus CO2. So the end product is ribulose 5-phosphate. What you gain, NAD plus is reduced to NADPH, so you get NADPH in this step. Remember, dehydrogenase plus CO2 means you are doing an oxidative decarboxylation in step 3. Okay, the net result from oxidative phase of PPP is from 1 glucose 6-phosphate, it produces 2 NADPH, 1 CO2, and 1 ribulose 5-phosphate. The net result of oxidative phase of PPP is 2 NADPH, 1 CO2, and 1 ribulose 5-phosphate. Then ribulose 5-phosphate can become ribose 5-phosphate for nucleotide synthesis, or enter the non-oxidative phase to shuffle sugars into glycolysis intermediates. Okay, so why NADPH from oxidative phase matters? Think of NADPH as protection and building power. So let's review it again. So it is involved in protection of RBCs. NADPH keeps glutathione reduced. Reduced glutathione detoxifies oxidants. So RBCs rely heavily on this because they have no mitochondria. And it also involves in respiratory burst. NADPH powers NADPH oxidase in neutrophils and macrophages. This helps generate reactive oxygen species to kill microbes. Another thing is NADPH also involves in biosynthesis of fatty acid, cholesterol, and steroids. Okay, and it is also involved in cytochrome P450 drug metabolism, which is detoxification. The USMLE questions usually test you about G6PD deficiency. If G6PD is deficient, you cannot make enough NADPH in RBCs. Then glutathione cannot stay reduced. Oxidative stress damages hemoglobin and the RBC membrane. So the triggers you should recognize are sulfur drugs, dapsone, primaquine, nitrofurantoin, or infections, or fava beans. The findings in peripheral smear would be Heinz bodies, which are damaged hemoglobin, and bite cells. When the spleen removes Heinz bodies, the RBCs become bite cells. So Heinz bodies and bite cells appear in G6PD deficiency in hemolysis. Alright, now we move to the non-oxidative phase of the PPP. This part is reversible and it is basically a sugar shuffling system. It lets the cell convert 5-carbon sugars into either ribose 5-phosphate for nucleotide synthesis or glycolysis intermediates such as fructose 6-phosphate and glyceraldehyde 3-phosphate. The big purpose of the non-oxidative phase helps the cell match what it needs. If the cell needs ribose for DNA or RNA, it can make ribose 5-phosphate. If the cell needs more glycolysis intermediates, it can convert pentoses into fructose 6-phosphate and glyceraldehyde 3-phosphate. In this non-oxidative phase, no NADPH is made. NADPH is made in only oxidative phase. The starting material for non-oxidative phase is the end material from oxidative 1. So the oxidative phase ends in ribulose 5-phosphate, a 5-carbon sugar. So from ribulose 5-phosphate, the cell forms 2 key 5-carbon sugars in non-oxidative phase. Number 1 is ribose 5-phosphate, which is used for nucleotide synthesis, and number 2 is xylulose 5-phosphate, which is used for carbon transfers. So remember, ribulose 5-phosphate can become ribose 5-phosphate and xylulose 5-phosphate. The 2 key enzymes in non-oxidative phase of PPP are transketolase and transaldolase. Trans-ketolase transfers 2 carbon units, and very important, it requires diamine pyrophosphate. It requires diamine, vitamin B1, and this is the classic vitamin hook of USMLE questions. Trans-ketolase transfers 2 carbon units. Trans-aldolase transfers 3 carbon units. It does not require TPP. It does not require diamine. So if the question mentions about diamine, the answer is almost always trans-ketolase. So the non-oxidative phase rearranges these 5 carbon sugars into glycolysis intermediates, fructose phosphate, glyceraldehyde 3-phosphate. These are standard glycolysis molecules, so this is how PPP connects back to glycolysis. There are 3 classic reaction sequences. Reaction 1 is by trans-ketolase 2 carbon transfer. So xylulose 5-phosphate and ribose 5-phosphate becomes glyceraldehyde 3-phosphate and pseudoheptulose 7-phosphate by the enzyme trans-ketolase, which requires the cofactor TPP, diamine pyrophosphate. So this is reaction 1. And the reaction 2 is trans-aldolase, by trans-aldolase, which is 3 carbon transfer. So this pseudoheptulose 7-phosphate plus glyceraldehyde 3-phosphate becomes fructose 6-phosphate plus erythrose 4-phosphate. And this reaction is by trans-aldolase. Okay, next reaction 3 by trans-ketolase again, 2 carbon transfer. Xylulose 5-phosphate plus erythrose 4-phosphate becomes fructose 6-phosphate plus glyceraldehyde 3-phosphate. By the enzyme trans-ketolase, the cofactor is again TPP. So as a net takeaway, if you can remember anything, just memorize this. The non-oxidative phase of PPP converts pentose sugars into fructose 6-phosphate and glyceraldehyde 3-phosphate. And it links PPP to glycolysis. And remember this concept, the non-oxidative phase can go in both directions. This is very important. Let's talk about some scenarios. Scenario 1. The cell needs ribose for DNA or RNA but does not need any DPH. Example in rapidly dividing cancer cell. In this case, the pathway can run backwards. It takes glycolysis intermediates, fructose phosphate, glyceraldehyde 3-phosphate, and converts them into ribose 5-phosphate. No oxidative phase needed, no NADPH produced, just ribose. Scenario 2. The cell needs NADPH but does not need ribose. Example, liver doing fatty acid synthesis. In this case, ribose 5-phosphate is converted back into fructose 6-phosphate, glyceraldehyde 3-phosphate, which re-enter glycolysis and can regenerate glucose 6-phosphate. That glucose 6-phosphate can go back into the oxidative phase again. So you can keep making NADPH over and over. This is called recycling. Okay? And remember, again, trans-ketolase from non-oxidative phase requires diamine. Diamine is vitamin B1. So in diamine deficiency, trans-ketolase activity decreases. This is classically tested in chronic alcohol use, Wernicke encephalopathy, in such scenarios. Sometimes exam stamps will mention decreased trans-ketolase activity in RBCs, and that's your clue. So let's summarize the non-oxidative phase. Location is in the cytosol. It produces ribose 5-phosphate. The enzymes mainly required are trans-ketolase, which needs diamine, and trans-aldolase. Is it reversible? Yes, it's reversible. And it connects pentose phosphate pathway to glycolysis via fructose phosphate and glyceraldehyde 3-phosphate. And it adjusts depending on whether the cell needs ribose, NADPH, or both. Now let's think about a question. If a rapidly dividing tumor cell needs nucleotides but not much NADPH. Which phase of the pentose phosphate pathway is most active? Answer is non-oxidative phase. Okay, another one. If a hepatocyte is synthesizing fatty acids and needs lots of NADPH, which phase is critical? Of course, oxidative phase, with recycling through non-oxidative reactions. Okay, let's conclude about PPP in a brief summary. So the pentose phosphate pathway, PPP, is a cytosolic pathway that branches from glucose 6-phosphate and has two main outputs, NADPH and ribose 5-phosphate. And it has two phases, the oxidative phase and the non-oxidative phase. The oxidative phase is irreversible and is where NADPH is made. The rate-limiting enzyme is G6PD, and this is the phase tied to RBC protection from oxidative stress and classic G6PD deficiency questions. The next non-oxidative phase is reversible and mainly shuffles sugars. It can generate ribose 5-phosphate for nucleotide synthesis or convert pentoses into glycolysis intermediates such as fructose 6-phosphate and glyceraldehyde 3-phosphate. The key enzymes are transketolase, which is TPP dependent, thiamine dependent, 2-carbon transfer, and transaldolase, which is for 3-carbon transfer. So the one-line memory is oxidative makes NADPH, non-oxidative rearranges carbons to make ribose or feed glycolysis. Okay, that's all for this podcast. Good luck to you all and have a very good day. Bye bye.